Good morning, everyone. Today we are going to get into a more detailed physiological lecture, and we will dedicate all our time to understand two special senses, vision and audition. As learning objectives, we will try to understand which mechanisms the animal use in order to see and to hear. As part of that general objective, we need to learn how sound, which is a pressure wave, can be converted to an action potential for the higher centers to understand. The same applies to light. We will elucidate how this type of signal is converted to an action potential. We will end up reviewing some basic ocular reflexes. Let's start with the sense of hearing. First of all, we need to figure it out: how do animals detect sound? In fact, this is not very complicated. It requires the external ear, which is the area encompassing from the pinna to the tympanic membrane. Sounds travel through the air in the form of a sound pressure wave, which is of different lengths depending on the frequency of the sound. These waves are picked up by the external ear and guided by the pinna, or the visible part of the ear, which is mobile and has a function of funneling the sound in. Once the sound has entered the ear, it triggers the vibration of the tympanic membrane. Here we can see how the sound is able to reach inside the ear and find a small barrier, which is the tympanic membrane, or what we commonly call the eardrum. Once it reaches the tympanic membrane, the sound wave is converted to a mechanical action because the sound wave physically pushed against the eardrum, forcing this conversion or transduction. This converted signal is also amplified within the middle ear. So now we do not have a sound wave, but we have a physical vibration of the eardrum, which is in contact with and transmitting the vibration to the ossicles. The ossicles are a series of three very small bones called the malleus, incus, and stapes, or what are more commonly referred to as the anvil, hammer, and stirrup, respectively. I repeat, all these bones are physically located in the middle ear, and their role is to convey and amplify this vibration from the eardrum to the oval window. Let's now look at the entire picture, looking at all the important components through which sound travels, followed by how and where the sound is converted to mechanical actions, and how this is eventually converted to an action potential, which will eventually reach the cerebral cortex to be recognized as sound. From the environment, the sound is initially guided to the ear through the vertical canal, which is supported by the auricular cartilage in the upper portion of the pinna. The vertical canal brings the sound down to the horizontal canal, which finishes at the tympanic membrane, which, as we mentioned before, vibrates at the frequency of the incoming sound waves. All of these constitute the actions happening in the external or outer ear. Then we encounter the middle ear cavity made by the bulla, and this area is connected to the auditory tube or Eustachian tube, which serves to equalize the pressure of the middle ear cavity with that of the atmosphere. This tube is the one you have to open by yawning when you are in a plane that is landing. As you get close to landing, the higher atmospheric pressure outside the ear with respect to that inside the middle ear causes that typical discomfort. You have to open the Eustachian tube to equalize the pressure. Attached to the tympanic membrane are the auditory ossicles that we mentioned a little while ago, which carry the converted sound wave into mechanical force and amplify this signal to stimulate the cochlea. The cochlea is the organ where the sound is eventually converted to an action potential. Let's magnify this section. Remember, the sound wave came through the outer ear to the tympanic membrane. From here, we have the middle ear, and we can clearly see the connection between the eardrum to the malleus, which connects to the incus, and this in turn moves the stirrup, which then physically pushes into the oval window to send the information into the inner ear. The next steps will sound a bit confusing because there are a list of events taking place in the inner ear, and we have not discussed those yet. However, the entire picture should become crystal clear towards the end of this section after we explain each step in detail. For now, let's follow the path of the signal. With that on mind, we find that the result of the stirrup pushing into the oval window is the vibration of the perilymph within the cochlea, which in turn will make the endolymph vibrate, and this vibration will make the basilar membrane to deform. The result of the deformation of the basilar membrane will be the eventual bending of hair cells, which are the sensory cells for audition. Bending hair cells will force the opening of potassium channels in hair cells, triggering the depolarization of these cells. The depolarization will become an action potential. The physical action of bending hair cells, which will result in the depolarization of these cells, mark the transition from a mechanical signal to an electrical signal in the form of an action potential. The action potential now will travel through the cochlear nerve to the medulla oblongata, continuing in the midbrain and the thalamus before reaching the auditory cortex, where the sound signal will be integrated and interpreted as sound. In order to fully understand the process of converting mechanical action into an action potential, we need to understand very well the structures in which the specialized cells that make the conversion are located, as well as the detailed descriptions of the cells themselves. All the structures which are defined as part of the inner ear are located in a cavity carved within the temporal bone. This is called the bony labyrinth, which in turn is divided into three functional areas. The semicircular canal, which are three tubular structures located perpendicular to each other, and they are involved in assessing rotational movements. The second component of the bony labyrinth is the vestibule, which evaluates linear movement. We will dedicate the next lecture to deal with these two components. The third structure of the bony labyrinth, and the one that we're interested in today, is the cochlea. Inside the bony labyrinth, we find a structure which mostly mimics the shape of the bony labyrinth. This structure is made of membranous tissue, which is separated from the bony labyrinth by a fluid called perilymph, which has the same characteristic as plasma and cerebrospinal fluid, which means that sodium is the main cation. The inside of the membranous tissue is filled with a different fluid, the endolymph, which has potassium as the main cation. If we make a cut in the tubular cochlea, we will see the different components. The outermost wall represents the bony labyrinths, within which, in the middle, we find the membranous labyrinth. The upper portion constitutes the scala vestibuli, to which the oval window connects in order to deliver the vibration coming from the stirrup. At the bottom, we find the scala tympani that ends at the round window. Both scala vestibuli and scala tympani are filled with perilymph, and they are connected at the helicotrema, or the tip of the spiral configuration of the cochlea. The scala media, located inside the membranous labyrinth, is filled with endolymph. The membrane which separates the scala vestibuli with the scala media is called the vestibular membrane, while the membrane that separates the scala media from the scala tympani is called the basilar membrane. Sitting on top of the basilar membrane is the organ of Corti, or the sensory structure of the cochlea. The organ of Corti runs the entire length of the cochlea, and it is made of several structurally supportive cells, in which 3 rows of outer hair cells and 1 row of inner hair cells are nested. The cuticular plate of the hair cells plus the surface of the supportive cells form an impermeable membrane called the reticular membrane, which permit that the stereocilia, or the hair bundle of the hair cells, be exposed to endolymph, while the body be exposed to corticolymph, a fluid with the same characteristic than perilymph. Sitting immediately above the stereocilia, we find the tectorial membrane, which play a crucial role in the process of depolarizing the hair cells to convert the mechanical action into an action potential. Here we can see an electromicrograph showing clearly 3 rows of outer hair cells and 1 row of inner hair cells. You may notice that all the surface is fully closed, making it an impermeable membrane. In this scanning electromicrograph, we can see the 3 rows of outer hair cells clearly defined, and we can see underneath the cuticular membrane the space where the body of the hair cells is bathed in corticolymph. There are some variation in the structure of different hair cells, but they all operate under the same principle. Let's look at the important part of an inner hair cell. Functionally, they can be divided into 2 main components. Several rows of finger-like projection of increasing height, called stereocilia, located in the cuticular plate at the upper part of the cell. These are always bathed in a medium containing endolymph. The other component of the cells is the body of the cell, which is bathed in corticolymph or perilymph. In the case of the cochlea, these 2 segments are separated by the reticular membrane. The shorter stereocilia is linked to a taller stereocilia by a connecting gating spring or tip link, which is a protein attached to the lower stereocilia tip and to a mechanically activated gated ion channel, specifically a potassium channel, inserted in the taller stereocilia. We will get into the function of this in a minute. A view of the entire cell shows the stereocilia at the top and the body in the lower part, and within the body we find potassium leak channels, voltage gated calcium channels, and then the ganglion cells which carry action potential to higher centers. As indicated before, there is a slight difference in the conformation of the hair cells in different organs of the body, but they all operate in the same manner. We just reviewed the structure of the inner hair cells of the cochlea. The outer hair cells of the cochlea have the same component, but the placement of the stereocilia in the cuticular plate is slightly different. You can see the conformation in here. The hair cells of the vestibule are also a different version. They have the stereocilia organized in a different distribution over the cuticular plate, but they also have a single tallest finger-like projection, which is called the kinocilium. The role of the kinocilium has not yet been identified as any different than all the other stereocilia. In this micrograph, we can observe the distribution of the stereocilia of vestibular hair cells. A close-up of the stereocilia shows clearly the connection between the top of the lower stereocilia to the taller stereocilia. Here we can see several of those connections. When the cells are not depolarized, the stereocilia is standing straight up. If the stereocilia bends in the direction of the tallest stereocilia or the kinocilium, the cell depolarizes and sends an action potential. A detailed view of the process is here. The stereocilia is standing up and the gating spring is not opening the mechanical potassium channel, so the cell is in its normal resting state. Once they bend towards the taller stereocilia, the gating springs open the mechanically activated gate ion channel, and that will trigger a depolarization. Upon returning to a straight position, the depolarization is terminated. When the stereocilia bends in the direction of the shorter stereocilia, then the cell hyperpolarizes, and then it comes back up and retakes its resting state. Hair cells are highly specialized cells capable of converting a mechanical signal into an action potential. There are several types of hair cells with slightly different configuration, but basically they all operate in a similar manner. The cells associated with equilibrium are located in the vestibule within the macula of both the utricle and the saccule, as well as those cells in the ampullae of the semicircular canal. The other type of hair cells which do not have a kinocilium are found in the organ of Corti, located over the basilar membrane along the length of the cochlea where they detect sound. The later version is described here. The hair cells consist of two main components: several rows of finger-like projection of increasing height called stereocilia, which are located in the cuticular plate at the tip of the cell, and the rest of the body. All stereocilia are based in endolymph, which is characterized for being a fluid rich in potassium, while the body of the cells is based in perilymph or cortical lymph, a liquid rich in sodium. The shorter stereocilia is linked to a taller stereocilia by a connecting gating spring or tip link, which is a protein attached to the lower stereocilia tip and to a mechanically activated gated ion channel in the taller stereocilia. When the stereocilia bends in the direction of the taller stereocilia, the connecting gating spring mechanically opens the potassium channel located in the taller stereocilia. This aperture permits the rapid uptake of potassium from the endolimbs, generating a more positive environment within the hair cells, which trigger the depolarization of the hair cells. The consequence of the depolarization is the opening of voltage-gated calcium channels in the body of the hair cells, causing an inward flow of calcium from the cortical lymph, which generating a transient elevation in intracellular calcium, which triggers the release of the neurotransmitter glutamate from a specialized vesicle at the base of the hair cells. Glutamate activates the ganglion cell to send an action potential to the central nervous system with the pertinent information. The signal is stopped when the stereocilia straighten up, causing the potassium channels to close. Now that we understand how a single cell depolarizes, let's see how does the organ of Corti functions in order to convert the mechanical action coming from the compression of the oval window into an action potential. A transversal cut of the cochlea shows several structures associated with audition. The scala vestibuli, which is separated from the scala media or cochlear duct by the Reisner or vestibular membrane. The organ of Corti, which sits in the basilar membrane, which in turn separates the scala tympani. The scala vestibuli and the scala tympani are connected at the tip of the cochlea by the helicotrema and are both filled with perilymph, which is a fluid rich in sodium. The scala media or cochlear duct is filled with endolymph, which is a fluid high in potassium. The organ of Corti is made up of 3 rows of outer hair cells and 1 row of inner hair cells. All hair cells are associated with a variety of supporting cells which in the upper part connect to the cuticular plate of the hair cells, forming a reticular membrane which is impermeable to the endolymph in which the stereocilia is bathed. Within the organ of Corti, below the reticular membrane, there are several aspects and spaces such as the tunnel of Corti, which are filled with cortical lymph, a fluid with similar characteristics than perilymph and to which the body of the hair cells are exposed. Within the cochlear duct, above the hair cells and in contact with the stereocilia, is located the tectorial membrane. Once the vibration is received by the oval window, that vibration travels through the scala vestibuli as pressure waves in the perilymph, which is transmitted through the vestibular membrane to the endolymph of the cochlear duct. This pressure causes the basilar membrane to oscillate, and the pressure is dissipated by the perilymph of the scala tympani once it reaches the round window. The oscillation of the basilar membrane changes the angle of the organ of Corti respect to the tectorial membrane. As a consequence of this, the stereocilia of the hair cells bend towards the taller stereocilia, thus opening the potassium channels and depolarizing the hair cells as the first step to generate an action potential in the ganglion cells, which leave the cochlea through the cochlear nerve, carrying the action potentials to the brain. After the pressure wave subsides, the basilar membrane returns to its resting position, permitting that the stereocilia straighten up and the hair cells no longer are depolarized, thus stopping the action potential. So far, we have figured it out. How does the sound coming to the outer ear in the form of pressure waves get to the eardrum, where it is converted to the mechanical action in the middle ear, and then this is eventually converted to an action potential by the organ of Corti in the inner ear. Now we should find out how we can recognize the different frequencies of the sound. We have the cochlea, and at one end is where the oval window, which is impacted by the stirrup, is located. At the other end, we found the helicotrema, which is the tip of the cochlea where the scala vestibula finishes and connects to the scala tympani. That is why both contain the same fluid perilymph. Remember that in the middle is the membranous component that contains the scala media and the organ of Corti sitting on the basilar membrane. If we uncurl the cochlea and make it flat, we realize that the width of the basilar membrane at the oval window end is much narrower than at the helicotrema end. The width of the basilar membrane determines which frequencies can deflect that section of the organ of Corti, therefore sending that frequency signal to the brain. The basilar membrane at the oval window, being narrower, can only be deflected by high frequencies. Therefore, the hair cells in this area can detect as high as 20 kHz. At the helicotrema end, the basilar membrane, being wider, can deflect the lower frequencies. Therefore, the hair cells in this area can detect as low as 20 Hz. The range we just described applies mainly to humans and to many other animals, but for example, some studies report that elephants can hear frequencies below 15 Hz, while other species such as the cat can hear frequency as high as 79 kHz. That is why they are able to find mice so easily, because mice communicate in the range of 1 to 100 kHz. Bats use frequency of up to 120 kHz for echolocation, and dolphins can hear as high as 150 kHz frequencies. You have a supplementary video available in Moodle to reinforce this information, but it would be too repetitive to show it here. To give you an idea of how these frequencies sound, I would like to do the following exercise. Starting at the low frequency, please hear 100 Hz, 300 Hz, 1000 Hz, 3000 Hz, 5000 Hz, 10,000 Hz. Usually, humans start losing the ability to detect the higher frequencies as age ensues. I sincerely hope that you can detect all these sounds that we just made. My ear, fortunately, is limited to a maximum of 7,650, so I didn't hear the last one. If you want to determine your hearing range, listen to a continuum scale I downloaded from YouTube and I am providing in Moodle, and you can determine what is your personal range. Finally, and for your personal pleasure, Please enjoy this wonderful piece. I hope this gives you a good idea of how the basilar membrane oscillates to generate the different frequencies that you hear. Now that we finished reviewing the basis of hearing, let's move to start reviewing the basis of vision. We will skip the microanatomy of the eye because you covered that in high school. We will start looking in some detail to the structure of the retina. After the sclera, which is the white of the eye, and towards the interior of the eye, we find several layers that all contribute to vision. There is a pigmented layer that contains the choroid, or the area where all vascular tissue is located and irrigates the eye from there. Also, pigmented cells which transfer nutrient and oxygen to the retina are located here. They are called pigmented cells because these cells contain a dark pigment which prevents excessive scattering of the light, thus sharpening vision. In this image, you can clearly see the sclera plus the pigmented layer with blood vessels and pigmented cells. Albino animals lack this pigmentation and, as a consequence, have a much poorer vision than other animals. Then we have what is described as the neural layer, which the specialist will be able to separate in 10 different layers. For our purposes, we will summarize this into the 3 most important components, which we will describe in the order that they go towards the lumen of the eye. First are the photoreceptors. This is a combination of rods and cones, and in here they appear as 4 layers, but in reality each of those layers is one part or section of each photoreceptors. Then we have a layer of bipolar horizontals and amacrine cells, and then the ganglionic layer made of ganglion cells which generate the action potential with the signal of the light towards the higher centers. Again, in here it appears to be 2 different layers, but they are parts of the ganglion cells with the bodies and the axon respectively. In the next 2 scanning electron micrographs, we can see the 2 types of photoreceptors, the rods, which make the majority of the population cells, and the cones, which are much less. Rods and cones. Now, going back to my cartoons of the neural cellular layers of the retina, we can observe the rods and the cones, which are the photoreceptors and have a clear outer segment, which is the photosensitive area of these cells, then an inner segment, which is basically structural support plus the equivalent of the guts or machinery of these cells, followed by the nuclear area, and finally the axons. Then we can see that the receptor cells synapse with the bipolar cells, which are regulated by horizontal and amacrine cells, although in this picture we do not see an amacrine cell. And these interneurons then synapse with the ganglion cells, which will eventually generate the optic nerve. In this image, we can see all the above mentioned layers in the eye. The light enters the eye through the pupil and first encounters the ganglion layer, then the interneurons, and finally reach the photoreceptors. All of these are facing the pigmented layer, which is sitting on top of the vascular layer. If we magnify this picture, we start seeing more clearly the position we see in the eye. If we eliminate the pigmentation, we can see clearly all the components here, including the amacrine cells. Now that we can recognize the main component of the visual system, let's elucidate how the action potential that conveys the vision to the brain is created. In this image, we can see the photoreceptor rhodopsin, which is made out of two components, an opsin, which in this case is painted in purple. In this case, that opsin is called scotopsin, which is a G protein coupled receptor embedded in the bilayer membrane of the photoreceptors. And you can see it crossing it several times. The other component of rhodopsin is retinal, which is this yellowish molecule. Okay? And this retinal is derived from vitamin A. That's why you have to eat your carrots. Of course, internally we have G protein and an intracellular cyclic GMP phosphodiesterase enzyme that play an important role in this system. The final component is a gated sodium channel, which is normally maintained open under darkness. The system to transduce light entering into the eye into an action potential, which convey this information to the brain, is as follows. Light is received by the retina entering through the pupil. The retina is made out of several types of cells, all located in the neural layer. These cells convert the input entered the eye in the form of light into an action potential, which is sent to the visual cortex for interpretation. In a counterintuitive manner, the light receptor cells, either rods or cones, are pointing towards the pigmented layer of the eye instead of towards the cornea. Both types of cells have an outer segment made of many discs which contain the photosensitive units. Towards the lumen of the eye follow the inner segment, the body of the axons of these photoreceptor cells. Their axons connect to bipolar interneurons, which are interlinked by horizontals and amacrine cells, which modulate the signal. Closer to the lumen still are the ganglion cells, whose axons form the optic nerve. In darkness, the photosensitive cells located within the disc have cyclic GMP attached to the sodium channels, and as a result, these channels are kept open, thus sodium is constantly entering the photoreceptor. At the same time, sodium is being pumped out of the cell by an ATPase pump. These situations maintain the membrane potential at about 30 millivolt, which imply to have these cells depolarize. While depolarized, these cells release the neurotransmitter glutamate, which inhibits the release of neurotransmitter by the next cell in the chain, the bipolar neuron, to the ganglionic cells, preventing the ganglionic cell from sending an action potential to the cerebral cortex. To generate an action potential, light entering the eye hit a yellow pigment called retinal, which is tightly associated to a molecule called opsin. Opsin is part of a larger structure called rhodopsin, which is a protein with 7 transmembrane domains plus retinal. The change exerted by the light on retinal causes a cascade of conformational changes in the rhodopsin, which in turn activates an intracellular G protein, which then activates a cyclic GMP phosphodiesterase enzyme. These enzymes moves and convert the cyclic GMP bound to the sodium channel into GMP. When the channel is bound to GMP, it closes its gates, preventing further entrance of sodium. Nevertheless, the ATPase pump continues pumping sodium out of the receptor cell, and by doing that, it hyperpolarizes the photoreceptor. While the photoreceptor is hyperpolarized, the secretion of inhibitory glutamate is reduced, thus not inhibiting the bipolar cells, which then are free to release the neurotransmitter, which stimulate the ganglionic cell to start an action potential which will travel to cerebral cortex with the information associated to the input in the form of light or color received by the eye. Now that we know how the action potential with the information about what we saw in the form of light was created, let's follow its path until it reaches the visual cortex in the cerebrum. For didactic purposes, I have separated the ball, which is our object of interest, into 2 balls. That will help to better explain what each eye sees, but in reality it is a single object. I have color-coded the different parts of the object seen and the pathways with the information. The information that enters the eye is always cross. That is, for example, the right eye can be divided into 2 sections. The temporal section, or right side in this case, which receives the information from the left side of the object, in this case the ball, and the nasal side of the eye, which sees the right side of the ball. The information travels to the brain through 2 pathways. The pathway followed by the information received by the temporal section of the eye travels ipsilaterally through the optic chiasm without crossing and reaching the right side of the thalamus. The thalamic neuron then sends the information through their axons to the posterior part of the brain in the right hemisphere of the brain. The information from the nasal side of the right eye sees the right side of the object, and this information travels to the optic chiasm, where it crosses to synapse in the left side of the thalamus, and from here to the visual cortex located in the left hemisphere of the cerebrum. The left side does exactly the same, but in the mirror image. Let's say the information about the right side of the object, seen by the temporal side of the eye, is sent ipsilaterally to the thalamus and then to the left side of the visual cortex in the left hemisphere. The information captured by the nasal side of the left eye, which is the left side of the ball, is carried to the optic chiasm, where it crosses to the right side of the thalamus, from where it is sent to the right side of the visual cortex in the right hemisphere of the cerebrum. The next topic that we have to deal with today has to do with pupillary reflexes. There is one reflex which you will have to constantly check as clinicians to determine the integrity of the neurons of both part of the central nervous system as well as parts of the peripheral nervous system. This reflex consists in the reduction of the diameter of the pupil when the retina receives an increased intensity of light. If there is no abnormality in the pathways of the information when one eye is illuminated, both pupils constrict. This is called a consensual response. The information received by one eye is processed in the midbrain, and efferent motor instructions go to both eyes to constrict uniformly. This reflex is used as a diagnostic tool to determine if there is proper sensory or motor function of the eyes, as well as to evaluate the brain stem function, because that's where the information is processed. If there is constriction in the illuminated eye, the right eye in this case, but not in the left, then most likely the problem is within the motor neuron of the left eye. The afferent signal is getting through. We see that because the right eye underwent constriction, but the motor component of the left eye is failing. Or there may be a problem in the brainstem before the signal goes out. Now, if we illuminate the right eye and only the left eye constrict, that indicates that the right sensory pathway is working properly, but the right motor neuron pathway has a problem. Now, if we illuminate the right eye and non-constrict, we know that either the right sensor is damaged or both motors have problems. Similarly, if we illuminate the left eye and none of them constrict, either the left sensor is damaged or both motors have problems. The last thing we have to review today is the electromagnetic wavelength spectrum that is visible. The spectrum goes from very low frequency, which is equivalent to very long wavelengths, to very high frequency, which is equivalent to very short wavelengths. Please do not confuse these waves used in vision to those used in sound. They are totally different. Here, you can visualize a wave produced by sound. The book hitting the table forces the medium to compress and to expand, and that is what carries the sound. Sound needs a medium to move along.
Electromagnetic radiation is a form of energy that propagates through space as oscillating waves that have electrical and magnetic components perpendicular to one another.
Electromagnetic waves, contrary to sound, does travel through empty space. That's why we can get light through space, outer space, coming to Earth. In the low frequency of the electromagnetic spectrum, we have radio waves, followed by microwaves, then the infrared spectrum, then we find visible spectrum, which goes from about 700 nanometers to 380 for humans. Dogs can see from 700 nanometers to about 360 nanometers, entering a bit in the ultraviolet spectrum. However, dogs do not seem to see the colors as humans do. They see everything in these colors. So they don't see red, and the yellow spectrum is much wider. Chickens do not see the red spectrum, but they appear to see a bit more into the ultraviolet spectrum. That's why you can use the red light at night to catch chickens, especially without disturbing them. Then we enter into the ultraviolet spectrum, followed by the X-ray spectrum, and finally we culminate with the atomic spectrum with their alpha, betas, and gamma rays. Lastly, and mainly as a curiosity for you, we have the tapetum lucidum. Tapetum lucidum is a reflector layer of cells located behind the retina in some nocturnal species that permit utilizing better the light in darkness, thus seen in dark environment. This reduces the sharpness of the vision, but increases the luminosity under which these animals can see. You can recognize a species with tapetum lucidum because they reflect the light when they are illuminated. This is all for today. It was a rather long presentation, but I will compensate for the next class with a shorter presentation. I made today's session a bit longer because I prefer to put all the subjects together, but if the volume does not allow me to load it as a single file, I may have to split this session into two components. To recap what we covered today, we basically covered the adaptations and component of both the ears and the eye to convert sound and light into common language that the central nervous system can use to interpret these signals. In specific, we covered the transformation of sound to an action potential and the conversion of light into an action potential. In both cases, we also reviewed the pathway from the receptor organ to the higher centers where these signals are interpreted. Finally, we described the ranges of frequencies that these two systems use to generate information that animals can detect, and mentioned a few species differences. On that note, have a good day.